Welcome. It is Saturday Night Live, broadcasting from the world headquarters of learn-biology.com in Berkeley, California. I am so incredibly happy to be here. And I see someone whose name is kind of like Phoenix, and that's fantastic. And I'm seeing a whole bunch of other people here too, and that is really wonderful. Sorry for the slight delay. There's always a technical problem or two. Hello, Sadhana. I hope I said your name correctly. It is fabulous to have you here with me tonight. Really, really great. So here we are, and I am going to start our show tonight. Here's tonight's agenda. First of all, welcome Biomanians, and that's who you are. If you are watching this show on a Saturday night, that says something incredibly special about you. It says that you're motivated and that your priorities are in the right place, that you are ready to do what we need to do in order to get to that 5 on the AP Bio exam. Sadhana, I am about to tell you that. Really, what we're going to do is focus on your questions. That's always been my top priority in these live sessions. But I did think that we would review Unit 4, which is a topic that I have neglected. And that includes some incredibly important stuff, including cell communication, homeostasis, feedback, and the cell cycle. It's fascinating stuff. But of course, I'm a biology teacher, so I think it's all fascinating stuff. So, Sadhana, I hope that was what you wanted to hear. And if it's not, you can ask me questions and we can really do anything. Because at this point, this close to the exam, it's really important that you get your questions answered. We're also, based on responses that I've received in previous sessions, we're also going to do some FRQ review. So again, I'm seeing more and more people enter into this live stream, and that makes me really happy because it makes me know that I am not alone in my love of biology, my favorite subject, and hopefully your favorite subject, despite the pressure that you might be feeling in relationship to the AP Bio test. And Phoenix, I hope I'm saying your name right because you've got a 3 in the middle of your name, but I'm going to talk about that right now. I do want to mention something. You're here, and I don't know, it's already 7 p.m. where I am, and it's probably either that time or later where you are, unless you're in Hawaii, and then it would be earlier. So here's the special offer. I, of course, am the creator of a website called learn-biology.com. It's a website that is designed to generate AP Bio success. And I'm encouraging people to sign up for it because in relationship to Phoenix's question, for example, it is by far the best way for you to review for the AP Bio exam. And I want to encourage you to sign up. It's not free. It's $24.95. You get complete access to the website. It's a monthly subscription, but you're going to cancel after the AP Bio exam. So that means that your total cost is going to be $24.95. If I have one person sign up, I'll do a little 15-minute session. If I get 2 people to sign up, I'll spread it out to 30 minutes. If I get 3, I'll do 40 minutes, 45 minutes. If I get more than 3, I'll stay on for another hour. So that'll be a very, very small group session, or maybe it'll be a big group session if a lot of you sign up. I see there's a bunch of people who are watching the stream. But what do you got to do? You got to go to learn-biology.com/AP Biology. All right, $24.95 for a monthly subscription. And you also get access to all the other live Zooms that we're going to do between now and the AP Bio exam. And somebody asked me about the schedule. If you type this in, learn-biology.com/live reviews, that's where you can see the schedule of the upcoming YouTube lives and the upcoming Zooms. We don't necessarily have a set schedule about what we do because at this point in the year, unlike previously, we're really following people's interests. So your questions can drive what we do in the Zoom sessions and in this session tonight. I am wondering just if we could do a little mic check. Folks, if the audio is okay and if you're hearing me okay, just put in the comment stream, sounds good, Mr. W, or even just sounds good. Okay, I'm seeing some talk about the APUSH exam, and I'm glad that that went well. Sounds good. Thanks, Phoenix. Okay, great. So, what do you do if you're getting ready for the AP Bio exam? Well, it's 5 days before the exam, and I just published a 5-day plan, and you can download it by typing in apbiosuccess.com/5-day plan. So no spaces, 5-day plan. apbiosuccess.com/5-day plan. If you do that, you'll get this. The front side is introductory. This is the back side, and there's a third page too. But basically, what it tells you to do is what to do every day. Now, it's not. It tells you what we're going to do every day, and basically you can customize it based on your strengths and your weaknesses. The thing to do, and this gets back to the question that came in from Phoenix, which is what do? You find your gaps. You find your gaps and then you fill them. You find your gaps in 2 ways. One is I've got a great checklist that you can download. I'll show you the link on the next page. You sort of go through that checklist and you're like, I know this, I don't know this, I know this, I don't know this. Then what you do is you fill your gaps with tutorials and flashcards, which are all available to you on learn-biology.com. Now is the time for interactivity. Don't do anything passive. You've got to be quizzing yourself and testing yourself constantly. And that's what the resources on learn-biology.com are all about. I've got hundreds of flashcards, hundreds of multiple choice quizzes, over 100 FRQs. And that's what you've got to do. You've got to test yourself, test yourself, test yourself. And you're constantly saying to yourself, oh, I know this, I'm good. No, I don't do this. I got to review it. I've got to deepen my knowledge. So that's it. It's this algorithm. Find your gaps, fill them. Find your gaps, fill them. Repeat that over and over again. Your study has to be active, and the way to go active is to go to learn-biology.com and go there. So the link here's you get the checklist once you go to our plan, and that's pretty much it. So again, I'm going to encourage you to do this a couple of times tonight, and I just told you that like you can get tutoring tonight that would cost hundreds of dollars, but you can get a session with me at 8:30. If you sign up for an account. So learn-biology.com, we've got tutorials, quizzes, interactive diagrams, games, comprehensive AP Bio exam reviews. It's what you need to get that 4 or 5 that you are craving. Any questions before we get into my plan, which is to review cell communication? Are there any topics that people wanted me to look at? So I'll just sort of take a deep breath. All right, well, you can ask me those questions at any time, So let's talk about cell communication. First thing is that just like you and I are communicating right now, living things communicate. Living things communicate constantly. And they communicate directly by touch. That is an animal thing and a plant thing. They communicate by signals, animal and plant thing. Bacteria do it too. But this communication by signals, that's the focus in AP Bio. And so the terms to know are this. Signals are ligands, and they bind with receptors. And that binding is specific. It's like enzyme and substrate. The substrate has a 3-dimensional shape that fits with the enzyme. Well, the ligand has a 3-dimensional shape that binds with the receptor. So it's a very specific connection. Binding leads to a cellular response, and that cellular response can be activation of genes. It can be activation of enzymes. There are 3 phases of cell communication that you need to know. And this diagram, diagram very much like it was on an FRQ a couple of years ago. And if you knew this, you'd be in great shape. So basically, there's reception of a ligand. And again, I'm drawing ridiculously simple shapes, but the idea is that the ligand shape is complementary to that of the receptor. If the receptor had a different shape, it wouldn't pick up this ligand. Then there's signal transduction. There's a signal that comes to the receptor, but then there's another message that goes into the cytoplasm and often to the nucleus. That change of the signal to another form is called a transduction. The verb is to transduce. So we're taking one signal, making it into another form. That involves second messengers and relay molecules, and then finally you get a cellular response. And that cellular response is either gene activation or enzyme activation. Now, an illustrative example of this are G protein coupled receptors. And they're important to know about because you're definitely going to get challenged with diagrams that are this complex. So that's one thing, just to like really contend with the complexity of AP Bio. But on top of that, what you need to know about is you'll see diagrams where they have things like kinases and stuff like that. So I want to see if I can make this a little bit bigger. Can I? Let's see. Didn't really work. We're just going to have to go with this as it is. Basic idea is this. In a G protein coupled receptor system, a signal, one common signal is adrenaline, because the way that your body responds to adrenaline is through a G protein coupled receptor system. Why is it called a G protein coupled receptor system? Well, here's the receptor, and next to it in the membrane is a G protein. So what happens is the ligand, adrenaline, that's that hormone that boosts your heart rate and makes your eyes dilate and sometimes gives people the ability to do feats of incredible strength. The ligand comes to the receptor. The receptor activates a G protein. So it's not a direct thing. This is the transduction that we talked about. The receptor activates the G protein. The G protein activates another membrane-embedded enzyme. That membrane-embedded enzyme in this case is called adenylyl cyclase. I swear that's its name. Unpronounceable, but that's the case. And what adenylyl cyclase does is its substrate is ATP, and it converts ATP to cyclic AMP. And cyclic AMP is ATP down to one phosphate, and the one phosphate is hung on to the molecule in a particular form. But the most important thing is that cyclic AMP is a second messenger, and it's a second messenger to know by memory. ATP, that's the energy molecule of the cell. One of the forms of ATP is cyclic AMP, and that is one of life's key second messengers. This system, by the way, is widespread in eukaryotes, and there are sort of analogous systems in prokaryotes. So pretty complicated so far what happens at the membrane. But now what we've done is we've transduced the signal. Now what we've got to do is we've got to get it deep into the membrane. So what it does is it activates a series of enzymes that are called kinases. Kinases are enzymes that become activated when they're phosphorylated. Phosphorylated, big terrible word? Not really. When you get a phosphate group, you're phosphorylated. So basically what happens is that cyclic AMP activates protein kinase 1, phosphorylating it. Now, what does protein kinase 1 do when it becomes activated? It activates protein kinase 2. What does that do when it becomes activated? It activates protein kinase 3, which then becomes activated, and it goes on in a very, very long chain. I can't really depict that here, but there'd be multiple chains that would be activated. And what you'd wind up with is signal amplification. You had one molecule of epinephrine or adrenaline, those are synonyms, coming in. And by the end, you'll have thousands of kinases that are activated. And what they'll do is they'll get to the terminal enzyme. And in this case, the terminal enzyme is called glycogen phosphorylase. And what it does is it causes glycogen in your liver to be broken down into glucose. That glucose streams into your blood. And what this is all part of is the fight or flight response. So you've activated the fight or flight response. You need energy to do that. That energy is in the form of glucose, streams into your blood. You run away, you fight back because you've been scared or something like that. And that's how this system works. On learn-biology.com, I have an entire tutorial about G protein coupled receptor systems, and it's worth knowing. I want to repeat my offer about anybody who signs up tonight can come to a Zoom review at 8:30 tonight. And I'm just going to play a moment of a really, I think it's great. I wrote it this year. And let me just get into the right screen over here. I think we're going to go like that. So watch this. Have you ever had a sudden fright? Perceived a threat, you're all geared up for fight or flight. Heart pounding, face flushed, hair standing up on end. It's partly from a hormone called epinephrine. Epinephrine's got another name, adrenaline. Secreted from adrenal glands into stuff in EpiPens, a shot can save your life. If you have an allergy involving sensitivity to nuts or sting from bees, epinephrine's polar, so it can't diffuse through the lipid bilayer. So instead, it induces a second messenger, it's cyclic AMP, which stimulates massive cellular activity. Let's learn how liver cells respond to an epinephrine dose. What they do is break down glycogen to glucose, which streams into your blood to give you energy required to respond to threats as you fight or flee. How does epinephrine get your liver cell to do this function? It's 3 steps: first reception and transduction, then cellular response and enzyme activation. We'll learn about the process of cell communication. Cellular communication works through phases 3. Reception of the ligand, they're complementary. Second messenger release, transduction is a beast. No amplified response until the stimulus has ceased. Today's focus is... Okay, enough of that. Thank you so much for listening. I hope you enjoyed that. And basically, it's 3 minutes, 4 minutes long, and it reviews all of cellular communication. I really recommend that you listen to it. You'll find it on the Science Music Videos channel. The hormones that we've talked about in every case, which cause a second messenger to be released, those are all polar hormones. And what that means is that they can't diffuse through the lipid bilayer. But there's another class of hormones that are steroid hormones, and they work by a different mechanism, and you need to know that mechanism. And so here's what you need to know. These steroid hormones can diffuse right through the lipid bilayer. And when they do, they bind with a membrane receptor, all right, a cytoplasmic, excuse me, I said membrane receptor, but what I meant is a cytoplasmic receptor. What does that mean? It's not bound to the membrane. It's not on the outside. It's actually floating around in the cytoplasm. Why can these steroid hormones make it through? They can make it through because they're non-polar. The phospholipid part of the bilayer is also non-polar. Like dissolves like. So these steroid hormones, examples are estrogen, testosterone. They can diffuse right through the membrane. They'll bind with a cytoplasmic receptor. They'll form a receptor hormone complex that will enter into the nucleus where it'll interact with DNA. Now, we're going to connect some stuff. We're sort of reviewing Unit 4, but now it's time to talk about the whole course. So this is a cytoplasmic receptor that becomes a transcription factor. And that means that it turns on genes. Turning on genes means production of RNA, which means ultimately production of protein through translation, through protein synthesis. As you pass through puberty just a couple of years ago, because I think most of you are probably 17, 16 years old, something like that. Well, puberty is about sex hormones that change your body. And those hormones include estrogen and testosterone. Excuse me, I got a little cord tangling here. Okay, estrogen and testosterone. And these hormones tend to result in long-term changes. When we were just talking about adrenaline and the release of the breakdown of glycogen into glucose, that's very quick. Your body needs to turn that on in seconds and turn it off in seconds. But this is long. This is long-term. And, think about the long-term changes involved in puberty. That's what it's all about. So steroid hormones tend to work more slowly. They involve activation of genes often, and that leads to longer-term changes. I see a question from someone who I'm meeting tonight for the first time, DSO World. Would calcium also be considered a second messenger when it is released from the smooth ER? It absolutely is. And in that case, usually what we have are nerve impulses that are acting as the first messenger. Those nerve impulses propagate themselves deep inside the cell, inside muscle cells. And those muscle cells have a specialized kind of endoplasmic reticulum that stores calcium. The calcium gets released, and that leads to the cytoplasmic changes that ultimately lead to muscle contraction. So the reason why I don't teach that is that's a little bit more connected to human physiology, but you are absolutely correct. Calcium is a second messenger, and you know, it's an important second messenger too. It's no more or less important than anything I've talked about tonight. That was a great question. Anybody else? Not seeing any other questions in the chat about anything I've talked about so far. Okay, let's go on. So, first topic in Unit 4, the first group of topics are all about cell communication. The second is about homeostasis and feedback. Homeostasis is one of the core concepts in biology. It's maintaining internal conditions at a relatively constant level. And feedback is when the output of the system feeds back into the system. It also becomes an input. And there's two types of feedback to know about. There's negative feedback. Negative feedback tends to quiet systems, turn them off. And then there's positive systems that accelerate internal changes and drive processes forward. You absolutely need to know, like, when a process is a negative feedback, when a process is a positive feedback. An important illustrative example, one that has a very, very good probability of showing up on the AP Bio exam, has many times in many years, is about insulin, glucagon, and blood glucose homeostasis. And the basic idea is that your body pretty carefully controls the level of blood glucose, the amount of glucose that's allowed to circulate into your blood. It keeps that constant. Why? The glucose is the fuel of your cells, so it can't go too low or else your cells would run out of food. But there can be many harmful effects connected with too much glucose in the blood, so your body's constantly regulating glucose levels. And in glucose homeostasis, we have two paired negative feedback loops. And here's basically how it works. The glucose set point is, I believe, something like 100 milligrams per deciliter, and that it doesn't matter. It has a set point, just like your body temperature has a set point. Body temperature set point, 37 degrees Celsius, 98.6 Fahrenheit. Well, there's a set point for glucose. All of these homeostatic variables have set points that they oscillate around. If you, for example, sit down and eat a bunch of mashed potatoes or a bowl of rice or a bowl of pasta or something really sugary, what that'll do is all of that, well, the starch will get broken down to sugar, or the sugar will directly diffuse from your intestine into your bloodstream. All of a sudden, you're above the set point. How does your body respond? An organ called the pancreas has cells that can detect the rise in glucose levels, and in response, they release the hormone insulin. Insulin goes everywhere in the body. The pancreas just releases it into the blood, but there are receptors for insulin in a variety of cells, including fat cells and liver cells. The liver cells do the opposite of what I just described in the phosphorylation cascade that leads to glucose release. What the liver cells do is they take glucose and they store it up as glycogen. Glucose is a monosaccharide. Glycogen is a polysaccharide. So they store glucose away as glycogen. And fat cells do a more complicated conversion where they'll take glucose and they'll actually convert it into fat. What's the result of that? Glucose is absorbed. Homeostasis is restored. As glucose is absorbed, the blood glucose level goes down. When it reaches the set point, the pancreas senses that, turns release of insulin off. Now, this system, which works to lower blood sugar level, is paired with another system that works to raise blood sugar level. Let's say that you've gone a couple of hours without eating. Well, you'll absorb all of the glucose in your blood, and your glucose level can't fall too low. Like, your brain will go into shock if the glucose level falls too low. So if you're below the set point, then the pancreas has other cells that release a hormone called glucagon. And glucagon does the opposite. It goes to the liver and it basically tells the liver, break down that stored glycogen and release it as glucose. As the liver does that, then glucose level rises, homeostasis is restored. So that, I would say, is worth committing to memory, understanding that feedback loop well. Now, and remember, those are both negative feedback loops. Why? The activity of the system is turning the system off. As we, for example, release blood glucose, as we release glucose into the blood, well, then the need to release glucagon gets turned off. And that's true of the release of insulin. As we release insulin, blood glucose levels go down, and then we can stop releasing insulin. Negative feedback in both cases. When this system breaks down, the result is diabetes. And in an FRQ or a multiple choice question, you might get the background information that would tell you, but if you know it ahead of time, you'll do better on the test because you don't have to figure it out from the reading. You know that, for example, type 1 diabetes is a disease where your pancreas loses the ability to secrete insulin. As to why, that's probably about your immune system attacking your own cells, which is outside the scope of the course. But know that type 1 diabetics, that used to be called juvenile diabetes, they can't produce insulin because there's a problem usually with their pancreas. Type 2 diabetics produce insulin, but their body's cells don't respond to the insulin signal. It's called insulin desensitization. So here you see insulin is still binding at the receptor, but the cells are not doing anything. Who does this happen to? It happens to people who are obese or are just overweight or sort of metabolically unlucky, but this is type 2 diabetes. And it's sort of a crisis in the United States because we have this obesity epidemic. So basically, if you eat too many carbohydrates all the time, your cells at a certain point say to you, are you kidding? More? I'm not doing it anymore. And that's what type 2 diabetes is all about. So questions about that? Not seeing any questions in the chat. Anything? Autonomy. You're asking me about what units we're going to go over in the next few streams. And I just want to put that back to you. I will always come prepared, but I'll also always make your questions the #1 priority. So again, remember how this review process is supposed to be working. You're supposed to be finding your gaps. How do you find the gaps? Use checklists, use the assessments that we have on learn-biology.com. The questions that you get wrong, that'll actually tell you what your gaps are. And then what you do is you do flashcards, you do tutorials, and then you test yourself again. So you can come to these sessions and say, hey, I was really having a lot of trouble with, for example, the difference between the leading and the lagging strand in DNA replication. Then we'll talk about that. Tyrosine kinase connector receptors, they're not dealt with as frequently as the systems that I've talked about tonight. This is a question from Deso World and or Disol World. I don't actually know how to pronounce that, but it won't hurt you to know about tyrosine kinase receptors. I don't teach it on learn-biology.com because I think it's a little bit too specific and it sort of falls outside of the scope of the course. I think if you know about G-coupled receptors, you'll be in good shape. And I think that if there was a question involving tyrosine kinase receptors, the College Board would give you the requisite background information so that you could reason your way through. So I wouldn't spend a lot of time studying that. My 2 cents. Okay, positive feedback. All we've talked about so far are negative feedback loops. Both insulin and glucagon work by negative feedback. What about positive feedback? Positive feedback is where the output of the system feeds back to the system to intensify its activity. The example that AP Bio students tend to be taught, and the one that you should know, relates to oxytocin and childbirth. So when a woman is pregnant, at a certain point, the fetus grows so large that it starts to stretch out the muscles that make up the uterus. In the same way as you can feel when your muscles are overstretched, there are stretch receptors in the uterus, and those are detected and they send a message to the brain. Um, I don't know how conscious that message is. It's not like when you try and touch your toes, but I also have never been pregnant, so I really can't speak with any authority about that. But the stretch receptors get activated. That sends a message to your brain where it gets picked up by a gland. I didn't list it here. It's not important for you to know. It's called the pituitary gland. The pituitary gland releases a hormone that's called oxytocin. Oxytocin circulates in the blood. It goes everywhere because hormones go everywhere, but they only get picked up by the cells that have receptors to them. The cells that have receptors to them include cells in the uterus, and oxytocin has the effect of causing the cells in the uterus, the muscle cells in the uterus, to contract. Wow. Well, more contraction, that means that there's going to be more activation of these stretch receptors. That's going to send a more intense message to the brain. That's going to lead to more contraction. Oh, more contraction, a more intense thing. And then that goes on until the baby is born. Congratulations, you have a baby. So that's really what that's all about. And Maxime, it is great to see you here tonight. And I am covering homeostasis. You missed cell communication, but we're going on and we're learning more and more because it is fabulous that all these people are here. And Pinita, I haven't seen you here in many nights, and it is great to see you here. I'm really, really happy to see you. Another hormone that works in the same way is actually unusually a gas. It's called ethylene. It's produced in fruit. So like if you imagine like a bunch of apples on a tree, or apples that you bought from the store and you bring them home, the first apple that becomes ripe, tissue changes within the apple cause it to release ethylene gas. That gets picked up by ethylene receptors in nearby fruit. As they become, that induces ripening in that tissue, and that causes those cells to release more ethylene, more fruit ripens, and so it becomes a positive feedback loop, and soon all of the fruit is ripe. Why is that a good thing? Well, it's an evolutionary thing because the purpose of fruit is to attract fruit eaters who eat the fruit and then poop out the seeds somewhere. Before there were people growing apple orchards, that's really what was going on. And that's a mechanism for seed dispersal. So it makes sense for the apples to coordinate their ripening. And this is how it's done, through this production of this gas. People have learned how to exploit this. So like, for example, the bananas that you eat are coming from places like Costa Rica and stuff like that. And they go on big container ships. And what they do is they make sure that ethylene is all pumped out, and usually carbon dioxide is pumped in. And Pinita, it was very nice of you to come by. And Kevin, I'm not really doing the review of the whole AP Bio exam at once because that would be like a 6-hour session, which I'm not up for. I'm up for this, though I am up for doing a little bit more tonight. And I've told people, I haven't seen anybody take me up on the offer. If you sign up for learn-biology.com, I'm going to put this in the comment loop. So if you go to the page that I just put in the comments, learn-biology.com/apbiology, it'll take you to, I'm just going to go there. I'm going to show you. Then I'll come back here. It'll take you to this page, and that shows you about all the features of the site. There's a little testimonial video that I think is great. There are testimonials from students. And then it sort of tells you about all the features, and you can learn about all the features of the site. And what you should do is call your parents over and say, hey, it's $24.95, and that will get me a subscription. And I encourage you to do that. And if you do that tonight, you can join us tonight for our special session of review. So here we are back here. Bam. Good. Okay. Oh, geez. That's just, I got a little bit ahead of myself, as I often did. So take your phone out, take a picture of the screen, show it to your parents. That's the deal. So Haven't seen anybody sign up for this yet, but again, if you do, you get another of the best review that you could get, because you can ask me any question or I can review any topic that you're interested in. All right, now let's talk about the cell cycle. It's not hard. You sort of need to know what happens in the cell cycle. You need to know it more for meiosis than actually for mitosis, but it makes sense to learn it in the context of meiosis. And I've got a great song about mitosis, and I'm going to take myself off screen, and you can listen to it while you get your parents to sign you up for learn-biology.com. So take a listen. Sign up. Guys, I want everyone's attention. We're going to go over what we did yesterday. We're going to go over the stages of mitosis. All right, everybody ready? All right, let's go. Interphase of cell division's longest part. Nuclear membranes intact as it starts. The cell's growing, cytoplasm flowing, chromosomes get duplicated, DNA gets replicated, chromosomes are spread out so they can't be seen distinctly, but note the nucleolus, the ribosome factory outside the nucleus, the two centrosomes. They later make a spindle which will pull apart the chromosomes. Prophase follows, the chromosomes condense, each is made of two sister chromatids like an X. Each sister is a clone, the closest of kin, and a centromere connects them like Siamese twins. The nucleolus disappears and melts away as the cell takes a ribosome production holiday. The centrosome separates, starts spindle formation for separating chromatids and cell elongation. Mitosis, chromosomal ride in a pro-meta-anatelophase divide. Eukaryotes go from one cell to two. Mitosis, how cells renew. In late prophase, pro-metaphase, the nuclear membrane. Okay, so that was fun. Mitosis, how cells divide into pro-meta-anatelophase divide. And again, just want to encourage people, call your folks over, have them sign up for learn-biology.com/AP Biology, and you get review sessions, you get so much. Hello, Esther. It's great to have you here tonight. So you definitely need to know about cell cycle and cell cycle checkpoints. So basically, there are three big moments within the cell cycle. Those are G1, generalized growth. S is for synthesis. That's when the DNA gets replicated. And G2 is the growth that is preparing the cell to undergo cell division. So you need to know those just like by heart, brute force memorization. I've got a tutorial about that on learn-biology.com. When that ends, that leads to the M phase, which is mitosis, the splitting up of the nucleus, followed by cytokinesis. Along the way, the cell is responding to certain internal signals that are monitored at checkpoints. A checkpoint is when the cell checks internal conditions. If the cell has met those internal conditions in a way that will lead to healthy cell replication, then the cell passes through that checkpoint. But if there's a problem, then the cell stops. So like, for example, if there are mutations over here, then the cell won't go into the S phase. Or if there's been insufficient growth, the cell won't go into the S phase. If the chromosomes are not looking good in terms of attachment to the spindle and stuff like that, that might stop G2. It's possible that that actually might be more relevant to the M phase. So the main thing to know is that there are checkpoints, and those checkpoints stop the cell at various moments in the cell cycle. The molecules that control that are cyclins. Those cyclins rise and fall during the cell cycle. You do not need to know the function of any particular cell cycle, but if you saw a diagram like this, you might think like there might be a question like, which cyclin peaks right before DNA replication? And you would have to think, oh, DNA replication happens during the S phase, therefore it's cyclin E. That's the kind of question that you would see related to that. Which cyclin might be connected with proper orientation of the chromosomes? It's probably cyclin B, because cyclin B is peaking right over here. So that's the way that cyclins are working. Now, in terms of actual control of the cell cycle, it's a little bit more complex. Basically, what happens is it's a combination of cyclins and CDKs. CDKs are cyclin-dependent kinases. We've talked about kinases before in the context of cell communication. Those are molecules that become activated by phosphorylation and then in turn phosphorylate other molecules. And this, like everything you learn in AP biology, as complex as it looks, is really complicated. I'm sorry. As complicated as this looks, it's really simplified. You know, when you go to graduate school or even when you major in biology, you'll see more of the reality. But even then, it's always simplified. So cyclins accumulate during the course of the cell cycle. They start out from a very low level, and then they build up and up and up and up. CDKs are at a constant level. When the cyclin concentration is high enough, they bind with the CDKs, and together they form a complex that's called MPF. It's called maturation promoting factor or mitosis promoting factor. It doesn't matter. You wouldn't have to really talk about what that is, but just know that this is this is basically what gives the cell the signal to go through the G2 checkpoint and then let the cell actually enter into mitosis. However, once that happens, the cyclin component of the MPF molecule will break down. So there's cyclin fragments, and then that will basically, after the cell divides, you know, and then we'd have two daughter cells, that sets the stage for the process to continue in each daughter cell. And I saw a question about what happens specifically during interphase. Sure. Interphase is when the cell doesn't look like it's dividing. It doesn't look like it's in mitosis. I'm going to just jump back here, okay? So during interphase, and if you were to look at, like I hope that you did the lab where you looked at onion root tip cells, most of the cells are in interphase. The cell is in the interphase most of its life. A lot of stuff is happening during interphase, but what's not happening is the nucleus isn't dividing. So you don't see distinct chromosomes in prophase, metaphase, anaphase, or telophase. That's not what you see. But what is happening during interphase? What's happening during interphase is this. 3 subphases. G1, the cell is growing. S, the DNA is replicating. G2, the cell is preparing to divide. So, Deso World's asking a good question. Does the MPF pretty much just phosphorylate mitosis proteins at the G2 checkpoint and then break down the cyclin? That is a really good way to think of it. I don't want to say yes, that might actually be a little bit more specific than you actually need to go to. But basically what MPF is this. In the same way as the cell is reading the levels of cyclins, it's also reading MPF. When you get to the G2 checkpoint, if MPF is present at a high enough concentration, that's the signal to go through the checkpoint. So Deso World, think of it that way, all right? When this concentration is high, that's the signal to pass the G2 checkpoint. And I would know this one. I don't know if you specifically need to know about mechanisms that are involved in other checkpoints. Again, there are tutorials about this all on learn-biology.com. Clicked in the wrong place. Okay. Now this is getting into some really cool territory, which is also AP Bio exam territory, because on the AP exam, they like to ask you questions either in FRQs or in multiple choice questions that relate to cancer. Why? Because AP Bio likes to ask questions about when systems that you've learned about break down. And cancer is a breakdown in the regulatory systems that we just talked about with cyclins and CDKs. So here's what you need to know. Cancer is caused by unregulated cell division, and it's caused by mutations in 2 classes of genes. The first are proto-oncogenes. Those are genes that under normal conditions promote cell division. You need cell division, right? That's how you grew from a single-celled individual to the trillions of cells that you are now through repeated cell division. You've got to have those genes. But when these genes mutate, they become oncogenes, and oncogenes cause unregulated cell division. They cause too much cell proliferation. There's another class of gene that are called tumor suppressor genes. And tumor suppressor genes are the ones that are responsible for the checkpoints. They say, no, 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 don't divide, don't divide. That's why they're called tumor suppressors, because when they're alive, when they're functional and healthy, that's when cells don't become cancerous. So in normal cells, you have tumor suppressors that are controlling cell division and growth factors that are acting at appropriate times. Those growth factors are coded for by the proto-oncogenes. When a cell becomes cancerous, it's kind of like the brake is broken. There's no brakes. You can't step on the brake. So the suppressors, the tumor suppressors, can't prevent cell division. And in cancer cells, the growth factor genes, those proto-oncogenes, they're saying divide, 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 divide all the time, even when it's inappropriate. I'm going to give you one in-depth example. This is a really cool example. It's called the ROS proto-oncogene. It's supposedly implicated in something like 30% of human cancers. And what's cool is that as an AP biology student, you know enough biology to understand how the system works. So when ROS is it's a G protein, which we just talked about earlier. So that's kind of cool. All these things are connected. As a proto-oncogene, ROS only becomes active when an outside growth factor ligand binds with ROS's coupled receptor. So we have the whole G protein thing. This becomes activated. It activates ROS. ROS would activate adenylyl cyclase. There'd be second messengers that would go into the cell. And those basically lead to a signaling cascade that result in activation of genes that lead to cell division. Okay? And Caton is asking a question about who has their AP Bio exam on Thursday. You must have a late administration of it, and that doesn't really impact what we're doing tonight. So this is ROS as a proto-oncogene. This is good, right? Cells need to divide. When they get signals, then they'll divide. All good. When ROS mutates, it becomes an oncogene, no longer a proto-oncogene. Now it's an oncogene. And look at what's happening. Even in the absence of a receptor being stimulated by a ligand, ROS is turned on. It's activating a phosphorylation cascade that's leading to the cell cycle. What that means is that it's become constitutively active. It's active by nature, okay? Active by nature. So it's constitutively active, and what happens in that case is this will bind GTP. That's how G proteins become active, even in the absence of a growth signal. Because ROS is always active, the growth factor is overproduced, and there's too much cell division. Too much cell division is what cancer is all about. So again, if you can sort of talk your way through diagrams like this and this, you have a good understanding of the cancer biology that you need to know for the AP Bio exam. Let's do some, okay, so we'll do one FRQ. That's really all we're gonna have time for. Though I would do more in this special session if people wind up signing up. So, and this is the special offer that I've made a couple of times, all right? $24.95, you gotta do it by about 8:15 because then I'll have time to, send you an email and the registration link, set up a Zoom, stuff like that. So you gotta do it, and again, don't do it for any other reason other than this is the way to get ready for the AP Bio exam. It's through interacting with the stuff we have on learn-biology.com. Let me just show you. This is a question from learn-biology.com. All right. So this one is not really about cell communication. It's about, I don't want to tell you what it's about. You'll see what it's about. So I'm just going to read it aloud. The plant is called Avicennia marina, is a species of mangrove that is adapted to grow in extremely salty conditions. Mangroves are often found in coastal waters and can withstand a salt concentration that's up to 2.5 times saltier than normal seawater. Avicennia marina has specialized gland which allowed the plant to secrete salt from the inner leaf to the surface of the cuticle. So you see that happening over here. Here's the cell, and there are all these proteins over here. And here's the direction of salt secretion. And you can see the salt over here on the outside of a leaf. So, two jobs. Explain the necessity of channel proteins in regulating salt concentration within Avicennia marina. Why do there have to be channels in order for salt regulation to be possible? And what is the type of transport as salt is moved from inside of the cell to outside of the cell? Justify your response. So, folks, class is in session. I'd like to see people like part 1. Channel proteins are necessary because. Who can give an explanation of that? I'll actually put a stem in the. If I can actually get my cursor over there. Good. All right. So I just put that in the comment thread. I don't know how long it takes for it to get there, but I wrote is channel proteins are necessary because. Why are channel proteins necessary? So I'd love to see an answer. Channel proteins are necessary because. Lily, I really appreciate, first of all, the fact that you're stepping forward. I'm way too big. There we are. There's one part of your answer that would lead you to lose a couple of points on this. Couple of points on this. You're doing great. And it actually relates to the second part of the question. But Lily, you're a hero. You're on the path. You're on the path towards a 4 or a 5 in the AP Bio exam. And Lily, I'm wondering if looking at part 2 and really looking carefully at the diagram, if you can think of a way to modify your response. Okay? Lily, take it all the way. Take it all the way, Lily. You can do this. If it's not diffusion, what must it be? And anybody else? We've got so many people. We've got Keaton. We've got Deso World. We've got Pranami Patel. So many people who could be chatting in. And you know the exam is right around the corner. You got to get yourself to this level. And that's really funny. Xialides. Xialides? I'm really sorry. You know, this is the weird thing. You could, if this were Zoom, you could tell me how to pronounce your name. Okay, so Lily, way to go. Indeed, it's active transport. So here is what you'd see on learn-biology.com. Basically, you'd click show me the answer. And the reason why I put this into the slide was just because the format didn't work so well, but this is an FRQ from learn-biology.com. Channel proteins are necessary because salt ions cannot move through the phospholipid bilayer freely because ions are charged. Therefore, channel proteins help move the ions across the membrane. But it's not facilitated diffusion, it's active transport because the cell is moving from inside to outside the leaf, and the ions are moving from an area of lower to higher concentration against the concentration gradient, and that requires energy. Okay. So let's do one more. We'll go a tiny bit over. This is pretty easy. This is right from learn-biology.com. Scientists wish to examine the effects of inhibiting ATP production on the transport of two different substances, A and B, across the membrane. Which substances transport will be most affected by the inhibition of ATP? And describe two possible reasons why A and B cannot move through the phospholipid bilayer directly. Anybody? Which substances transport will be most affected by the inhibition of A and B? We can actually treat this like a multiple choice question, A or B. Put it in the chat, everybody. Which substance is going to be more inhibited by the production of ATP? ATP. Simple as 1, 2, 3. And proud of me. Nice. I'm seeing all of these A's pop through, and you're all exactly right. And that's because A is obviously an active transport process where things are moving from low concentration to high concentration. So it has to be driven by active transport. Folks, if you sign up right now, I'll take you through 2 or more real FRQs. And let's just go here. All right, we're not going to be able to do this tonight. Want to repeat my special offer. Okay, so, call your folks over and just say, hey, this website is going to really help me on the AP Bio exam, and Mr. W is going to possibly tutor me. So, but you got to do it right now. You got to do it by 8:15, or, I'm going to do something else. So you got to do it by 8:15, and I wish I could leave this up while leaving the Zoom, but actually here's what I'm going to do. I'm just going to leave this up, and I'm going to take a tiny break, and I'm hoping that I'll see a couple of emails show up, and then I'll come back, and then we'll say good night. So here I go like this. Okay, folks, thank you so much for joining me tonight. It's been really fabulous to have you here. Again, sign up for learn-biology.com. You get these free Zoom reviews. You get the best website in the world for mastering AP biology. And you get support from the website 24/7. You get the interactivity that's going to allow you to do what you need to do. Oh, and you get the Biomania AP Bio App. You get great review in your pocket 24/7 between now and the AP Bio exam. I want to say thank you to every single one of you. I know you have many choices for what you do on a Saturday night, and you chose to spend an hour of it with me, and that was really awesome. So everybody, be well. Go to learn-biology.com/live-reviews, and that's where you'll see the schedule for the next ones. And again, thank you all so much for being with me tonight. Everybody, be well, take care of yourself, stay safe, study hard on learn-biology.com. learn-biology.com, the website that guarantees your AP Bio success. Thank you so much.